Hello guys, today we're talking about staghorn ferns. Well, what is a staghorn fern? Well, the staghorn fern is a type of fern that is epiphytic. Epiphytic means that it grows on trees rather than soil, but it does not mean that it is a parasite. It uses trees purely to support, drawing water and nutrition from rain, air, and fallen debris. This way of life makes the staghorn fern an expert at surviving in high canopy level environments where sunlight is more abundant but soil is absent. Well, let's talk about its habitat. Staghorn ferns are native to tropical and subtropical forests around the globe. You can find them clinging to tree trunks and branches. Have you ever wondered how staghorn ferns survive high up on tree trunks or rocky cliffs, far from any soil or regular water supply? These unique plants have evolved some fascinating strategies to store and transport water and nutrition. Well, first, let's talk about water storage. Staghorn ferns have shield-shaped leaves. Well, they do more than just look cool. They help reduce water loss and trap organic matter, creating a mini compost pile that boosts nutrition's availability. Even the little tiny white hairs on reproductive structures called strobili slow down evaporation, like natural sunscreen for the plant. And finally, the meaty leaves can store both water and nutrition, helping the fern survive in harsh, sun-scorched environments. Now, how does water move through the plant? Like most vascular plants, staghorn ferns absorb water through a simple fibrous root system. The water travels upward via xylem tissue, a hollow tube that is made of long dead cells, which is called tracheids. These structures have tiny pores that allow water to move between cells and their strong, lignified walls. It is like a one-way highway that water flows from the root to leaf, driven by evaporation at the top, creating a negative pressure. For transporting nutrients like sugars, ferns use the phylum. This includes the sieve cells, which are alive but lack a nucleus, and their helpers, the companion cells. Let's take a look at glucose. Glucose is actively pumped into the phylum, and also water follows by osmosis because the increased concentration of glucose, and so on, pressure builds. So this pressure pushes sugar throughout the plant. Unlike water, this system can flow both ways. So moving nutritions in plants is easy and convenient, so the nutritions can move to where they need the most. From water-saving leaf designs to ancient vascular highways, staghorn ferns are master survivors, living proof that evolution doesn't always mean complexity, just clever adaptation.
So I'm going to first talk about our first experiment, which is different light exposure. In this experiment, our objective is to see how different levels of light exposure influence plant growth. So, subsequently, with this experiment, we will be able to conclude which level of light, whether it is high, moderate, or low, are most efficient for the plant to grow fastest. pH conditions of the soil were also monitored in this experiment, although this wasn't the primary objective and was just a secondary thing that we did. pH testing is something that we did continuously to the end of our experiment, throughout the experiment too. So, for the tools used for the first experiment are plant growth light, timer switch, cardboard boxes, and rubber bands. So, the timer switch is used so that the plant growth light can be turned on and turned off on specific time. And the cardboard boxes are used to hold the plant growth light in place. And the rubber bands are used to tie the lights onto the boxes. We also had calipers, which are used to measure the growth of the plants, and pH testing paper, as well as syringe for the pH testing. And for the materials, we had plant pots and medium, as well as the silver staghorn fern plant, and we have 9 of each. And over here is a picture of our tools. There is the plant growth light on the very left, and then there is the timer switch and the lacquer paper. The experimental method right here, as I have mentioned, we exposed the plants onto different light levels. So the first group A, B, and C, are exposed to 12 hours of plant light every single day. And the second group D, E, and F, are exposed to 6 hours of plant light every day. The last group G, H, and I, are exposed to natural sunlight, and it is put next to a window. And we do our measurements every 7 days, and we observe the growth as well as measurements on the plants. We measure the length, width, and thickness of the leaf. And we also do pH testing for the soil, where we add a controlled amount of water and we take the water droplets from the pot and use it on the lactose fever, so it will indicate the pH of the overall environment. Here is a picture of our experimental setup. On the very left is our box setup for the plant light, where the plant is going to spend most of its time. And on the middle is a picture of how the methods of measurements are done. And on the very right is a picture of our pH testing example.
Next, the angle experiments discussed whether different angles impact the plant's growth. We control a 12-hour light exposure and 45 millimeter watering each week. We divided the plants into 3 groups. Each group have a specific angle facing the light sources. As we can see in the picture, the 3 angles are 0, 45, and 90 degrees. Just like the previous experiments, we water measure the growing status and test the pH rate each week.
So the next part, I will be presenting our findings on how daily light exposure affects the growth rate of silver staghorn fern. We compared 3 light conditions: 6 hours of artificial light per day, 12 hours of artificial light per day, and the natural sunlight. Over 5 weeks, we checked growth rate to identify the most effective photoperiod for cultivation. To ensure reliable results, we divided 3 ferns into 3 groups. The first group received 6 hours of LED grow light daily. The second received 12 hours, and the third was placed outdoors under natural sunlight, which varied depends on the weather. We measured growth weekly by calculating the percentage increase in frond length and counting new fronds. Now, let's look at what the data revealed. Here's the growth rate trend over 5 weeks. The y-axis shows growth percentage and x-axis tracks time. Starting with 6-hour group, growth was slow but steady, reaching 57% by week 5. This suggests that 6 hours of light is sufficient but survivable, but limits full potential. The 12-hour group had a dramatic spike of 448% growth in just week 1, like during the initial light saturation. However, by week 5, it dropped to 168%, possibly due to the stress or nutrient depletion. The sunlight group showed minimal growth, peaking at just 5%. We suspect environmental factors like temperature swings and a strong wind in Hsinchu affect the growing progress. In summary, 12-hour light boosts growth rapidly but require careful monitoring, while 6-hour light provides more stable growth. And under natural sunlight, the growing rate is most stable, but at the same time it is slow at growing. So first, the 12-hour photoperiod early suggests that silver staghorn ferns can exploit abundant light initially, but subsequent drop implies need for adjusted care, perhaps supplement nutrients or CO2. Second, although the sunlight is natural, uncontrolled conditions like winds or rapid temperature changes may do more harm than good for the species. To wrap up, our experiment highlights that photoperiod profoundly impacts silver staghorn fern growth.
While 12-hour light delivers rapid results, it demands attention. 6-hour light is slower but reliable, and natural sunlight may need human intervention.
Based on our analysis of the light exposure experiment, we can conclude that the most significant growth was recorded in the group exposed by the UV light for 6 hours a day. And it is followed by this 12 hours light group where the direct sunlight has the least growth changes. And the direct sunlight caused the fronds to become scorched or dry and crispy at the edge due to the overexposure. But conversely, the plants exposed by the low intensity of light became laggy, with fronds stretching towards light source. And based on our observations, we can conclude also there that the plants with the indirect light, such as 6 hours UV light, can have the optimal development of the optimal growth development. And we have the second experiment, which is the planting angle. Based on our experiment, we can observe that the growth of each plant was inconsistent. Since growth changes is inconsistent, we can specify which angle that has the most growth back to the silver staghorn fern. In the other hand, we have also the watering effects that affect the growth of this silver staghorn fern. The plant is sensitive to both overwatering and the underwatering. Overwatered plants will overcome the yellowing and blackening of the fronds, and also the symptoms of the root rot or fungal stress. But in contrast, the watered ferns showed drooping fronds and slowed growth, as the plant struggled to maintain the moisture balance. The results emphasize the well-regulated watering schedule, and we ensure the consistent moisture of the to avoid waterlogging the root system.